Okay, so we're going to move on now to our final speaker of the evening, and that is Dr. John Poole from the University of Wisconsin.
All right, hello, and thank you very much for being here at the end of a long but very satisfying day of science. So the story that I'll tell you today is kind of a two-part story. The first part has to do with our search for an efficient trait mapping method for Drosophila or other species with similar experimental properties. And the second part is an application of that related to our interest in the genetics of adaptive population differences in Drosophila. And before I go any further, I would like to emphasize that the second part in particular was led by Dr. Eloise Bastide, a recent postdoc in my lab, with help from additional lab members as well. So I'll start out by telling you about the bulk segregant mapping approach that this effort has led to. And in the second part of my talk, I'll tell you about applying that method to the genetic basis of pigmentation differences between Drosophila populations and connecting that with some population genomic data as well to give kind of a snapshot of what we found about the genetics of adaptation. So as you may be familiar with, bulk segregate analysis, at least as I'm going to describe it today, involves a cross between two phenotypically contrasting strains. We do both reciprocal crosses here, mix the F1s, allow them to interbreed at a fairly substantial population size for a number of generations, and then we select on a trait only at the end of that process. And we select the highest and lowest individuals in some proportion to separately sequence as pools. And why might we consider this? Well, it's a relatively small number of DNA libraries to prep if we're doing genomic sequencing, and or to genotype, however. And also, one thing we like about this is that we're often interested in multiple traits that may differ between a pair of Drosophila populations. And with this approach, we could potentially select on a different trait every generation and map multiple traits using the same experimental cross. At least if you're comparing the two tails, even if there's been some selection in the laboratory population, the worst that that's going to do is to reduce your power somewhat. So before I got into actually putting together this approach, I did some preliminary simulations. This simulator actually became the nucleus of the inference method that I'll talk about later. But initially, I just used it to try to define some basic experimental guidelines for this approach. For bulk segregate analysis. So I simulated the recombination of all the individuals in the mapping population, the selection on phenotypes with genetic environmental variants, and the sequencing, the sampling of reads from each pool. So I did that for bulk segregate analysis and also for integration mapping. And to make a long story short, because I don't want to dwell on this preliminary part of the story, bulk segregant did better in each case that I investigated, which was a large number of parameter combinations. Some of the conclusions of that analysis were quite obvious. More individuals is better, more generations can be better, more sequencing depth can be better. And this all kind of goes, behind maybe one of the golden rules of QTL mapping, which is that, one important goal is to sample a diversity of recombination breakpoints. So that's pretty obvious. And one thing that was maybe a little bit comforting was that there seemed to be a broad optimum for the proportion of individuals selected in the tails. But the first rule seems to be, avoid nasty sampling variance, and only then worry about selecting the most extreme individuals. Bulk segregate analysis has been applied very successfully in the yeast world, where you can generate millions of segregants. And this tends to yield sharp QTL peaks that are often very distinct. But in our case, now we can generate maybe 1 million offspring, 1,000 offspring, possibly more, but not 1 million. And so as a product of that, our QTL peaks may often be somewhat wider and may often overlap, at least if we're trying to map a complex trait. And that gets at the reason that I actually went about putting together a new approach for analyzing our data. I would have been quite happy if I didn't have to put together a new approach, but the existing methods, although they think they work very well for the data that they've been applied to, don't necessarily account for the possibility that QTL peaks may overlap. So that was the main motivation for putting together this approach that I call SIBS-SAM, which is, again, based on simulations of the experimental process. And so SIBS-SAM is aiming to make inferences about not only primary peaks, but also secondary peaks, which may have overlapping mapping signals with the tallest peak in the region. And I'm summarizing this based on what I call ancestry difference, which is basically the difference in the parental strain ancestry between the high phenotypic pool and the low phenotypic pool that were selected. So this is a scary looking flowchart, but it actually boils down to just a simple 3-step simulation-based process for making inference about QTLs from our bulk segregant experiments. The first step, the null simulations, are just what you'd think. They're simulations without any real QTLs, so all the phenotypic variance is just random environmental effects. And here we're looking to see if any of the peaks we observe in the real data are taller than what we would expect under that null hypothesis. That gives us a p-value for each primary peak. Okay, so for any primary peaks that are deemed significant, we go ahead and do single QTL simulations. That's the second step in the center column there. And in our single QTL simulations, our null hypothesis is that there's really only one QTL here. It's the one that we've already inferred. And our alternative hypothesis is that there's actually another one here as well. And that helps us to infer significant secondary peaks. But through a rejection sampling approach, we can also make inferences about the effect sizes and the genomic confidence interval around the primary peaks as well. Finally, if there are significant secondary peaks, then I do cluster simulations, so multi-QTL simulations, to try to clean up our inferences about each peak's effect size and location in light of the fact that they influence each other's signals. So that's the basic idea. This is a rather modest experimental design where we're only phenotyping 600 flies. But even here, at least we have good power to detect major effects, things with maybe more than 15% effect size. And in this context, it works pretty well as long as we're looking at QTLs that are more than about 10 centimorgans apart. It also estimates the effect size pretty well, but with a couple of exceptions shown here. The point on the left is a little off because of the Beavis effect. If you detect it at all, it's probably a little taller than average. And on the right, there's kind of a saturation effect where if you're explaining 50% of the variance, the statistic is kind of maxed out. It would be similar if it explained 80% of the variance. But at least for the intermediate effects, things look pretty good. All right, so moving on to the application of the approach, we're interested in this pigmentation system where, compared to the normal phenotype you see on top for Drosophila melanogaster, there are a couple of populations, including Cameroon and Uganda, that have evolved abdominal melanism, and a high-altitude Ethiopian population which has evolved a more extreme phenotype. So we applied our approach to a series of crosses between these different melanic populations and a light population from Zambia. This is one example result, plotting ancestry difference along the 5 major chromosome arms in Drosophila melanogaster in these different colors. And this result from a Uganda population is kind of a positive control. in that we already knew about the tallest peak there in red, which corresponds to the ebony gene, and that's something we'd even characterized at the level of mutations in a previous study. This is a bit newer result. This is looking at our highly melanic Ethiopian population. And again, one of the two tallest QTLs lines up over ebony on the right. The other lines up over tan, another well-known pigmentation gene on the left. And there are some minor secondary peaks that don't necessarily all correspond to known pigmentation genes. Only one of them does, in fact. I'm not going to focus on the potential novel loci here. I'm going to focus on the well-known pigmentation genes, and I'll show you why in a moment. But as far as what we learned from the QTLs alone, before I move on to the population genomic data, we saw a pattern of major effects in every cross that we looked at, at least moderate to large effects, I should say. And often we were detecting, more than a couple, but not, large enough effects that it does not appear to be highly polygenic, at least as far as we can tell. There were some interesting patterns that emerged in terms of the overlap of QTLs between crosses. There was evidence for some parallelism between populations, where the same QTL appeared in, let's say, an Ethiopia cross and a Cameroon cross. 6 out of the 18 QTL regions were found in that context. But interestingly, there was a fair bit of variability even between two crosses from the same population looking at the same trait. And so this may reflect, in addition to, random results based on limited power for weaker QTLs, this may also reflect pigmentation differences that are not fixed between populations and potentially epistasis on top of that. So moving on to the population genomic data, our prediction would be that if a gene was responsible for this recent adaptation, that it should have a population genetic signal of local adaptation. And we had these QTL peaks that often lined up over well-known pigmentation genes. In the last mapping plot that I just showed you, we had nice QTL peaks over ebony, tan, and yellow, which are probably the 3 genes most often implicated in Drosophila pigmentation evolution. And so it would be pretty devious of nature to have pigmentation be due to 3 other genes right near ebony, tan, and yellow. But intriguingly, there was no strong population genetic signal, at least at the window scale of roughly 5 kb windows in terms of genetic differentiation between the light and the dark populations. So what's going on here? Well, one clue is that when we zoomed in at the single SNP level at these genes, in decent sample sizes of, let's say, 30 to 50 individuals for Ethiopia, 200 genomes for Zambia, there were a small handful of SNPs. For example, this little cluster at ebony, which showed very strong allele frequency differences between populations, and yet without a very striking linkage signal at all. And similar patterns were observed at other loci, and we've observed similar things for other traits as well. So you actually get kind of a stronger signal, if you're looking at either comparisons to a neutral demographic model or comparisons to the genomic distribution of FST. You get a stronger signal if you look at just the maximum SNP FST in the window than you do if you look at the whole window's FST. Just a very narrow pattern of high genetic differentiation. And one reason that we might expect that is if we had something a little different than the classic hard sweep model. So a classic selective sweep model involves a new mutation on a single haplotype that selection boosts in frequency, and so you get strong linkage effects on each side of the beneficial mutation. On the other hand, if you have a soft sweep from standing genetic variation, that variant may have already been on more than one haplotype background, and so when selection raises it in frequency, the effects on the frequency differences of linked variants are much less pronounced. And so we used a simulation approach to explore whether that kind of model could explain what we're seeing at Ebony, using that as an example. And we actually got the best fit to the data in terms of the combination of a very strong SNP FST and a milder window FST for models of soft sweeps with relatively higher initial frequencies, which is compatible with the fact that at the most differentiated SNP, that the Ethiopian allele actually is at a frequency of roughly 3% in Zambia, the light population. So to summarize the talk, we did some simulations to kind of test out bulk segregate analysis as a mapping approach for Drosophila, and put together an inference approach based on these simulations. And investigating pigmentation as an application, we found that there were a fair number of loci of at least moderate effect, with kind of an intriguing pattern both within and between populations. And that zooming in on Ebony as one major effect locus in Ethiopia, we found evidence that genetic variation and phenotypic variation here may have been driven by selection on standing genetic variation. So lastly, I'd just like to thank the co-authors from my lab and funding, and certainly the organizers of this meeting, and PEQ especially, and thank you for your kind attention.
Okay, this was a wonderful session. Please join me in thanking all of our speakers again.